we could see some howling. So could you turn off the microphone?
Okay, but actually, we don't turn on our microphone.
Oh, that's so?
Yes.
Okay, then let's start anyway.
Hi, so good morning, everybody, and hello, guys in Taiwan side. Can you hear me? Okay. So I'm Kentaro Ifuku from Graduate School of Biostudies, and I'm going to talk about the evolution of oxygenic photosynthesis. So here is today's content of my talk. I suppose not everybody here is plant scientist, so I'd like to start my lecture with basics about photosynthesis. In particular, I will talk about the evolution of photosystems, photosynthetic reaction center. And then I'd like to talk about the acquisition of photosynthesis by symbiosis. And finally, I will talk about the protection mechanism of photosystems against the excess light environment. So let me start the first part. As you know, photosynthesis is a process converting light energy to the chemical energy. And this process can be divided into two processes, so-called light reaction and dark reaction. And the light reaction is a photosynthetic electron transfer, producing chemical energy like NADPH and ATP. On the other hand, dark reaction is an assimilation process, fixing carbon dioxide mainly, and also nitrogen and sulfur are fixed by the chemical energy produced by light energy. Then we animals use carbohydrate and oxygen produced by photosynthesis and obtain the chemical energy. And finally, chemical energy is dissipated as heat. So light energy is finally dissipated as heat. So that means the energy flow from the sun is one way, while the materials is cycling on Earth. And oxygenic photosynthesis is a driving force for material cycling on Earth. Then let's think about the evolution of oxygenic photosynthesis. This slide shows the rise of oxygen level on Earth during the early history of life. It is suggested that the starting point of oxygenic photosynthesis is around 3 billion years ago. And because of the fossil record of ancient cyanobacteria. And before this point, there should be divergence of non-oxygenic photosynthetic bacteria. So they use natural reducing chemicals such as hydrogen sulfide as an energy source. And before this point, there should be ecosystem without photosynthesis. So the question is, how is the life without photosynthesis? Even now, there is an ecosystem without photosynthesis, like a hydrothermal vent in deep seas. In this ecosystem, chemosynthetic bacteria obtain energy from natural inorganic chemicals. And there are some bacteria that have a bacterial type chlorophyll that absorb the infrared radiation. Lower panel shows the absorption spectra of bacterial chlorophyll and also the absorption spectra of chlorophyll A and chlorophyll B from green plant. You can see that bacterial chlorophyll absorb the light wavelengths around 800 nanometer to 1000 nanometer. So this suggests that bacterial chlorophyll might be a sensor to detect the heat in this dark ecosystem. And this would be the original function of chlorophyll pigment, photosynthetic pigment like chlorophyll. Then let's think about the oxygenic photosynthesis again. As I told you, oxygen photosynthetic start around 2.7 billion years ago. Because oxygen is very toxic to non-photosynthetic organisms. The unoxygenic organism is killed by the oxygen generated by the cyanobacteria. And this event is called as a great oxygen event and also oxygen catastrophe or oxygen crisis. However, life can overcome this problem and develop the aerobic respiration to get a big amount of energy. And this allows the emergence of larger eukaryote cell and also emergence of eukaryotic phototroph via symbiosis, and finally emergence of multicellular organism. And further increase of oxygen in our atmosphere allows the formation of ozone layer, and this allows the plant to extend their living area to the land surface. So this is a story of the evolution of oxygenic photosynthesis. And this cartoon shows the electron transport on silicon membrane of chloroplast. In silicon membrane of chloroplast, there are two types of light-driven energy conversion system called photosystem 2 and photosystem 1. And photosystem 2, in short, PS2, catalyze the water oxidation reaction and reduce the plastoquinone as a mobile electron carrier. And this mobile electron carrier, plastoquinone, is oxidized by the cytochrome B6 complex, and then electrons are transferred to PS1 and via plascyanin, and PS1 oxidized plascyanin to reduce ferredoxin and NADPH. And this NADPH is used as an electron source for carbon fixing reaction. And during this electron transfer process, protons are translocated into silicon lumen, and these protons create a protein motive force to make the ATP at the chloroplast ATP synthase. So the question is how these electron transfer systems have been evolved during the evolution. Interestingly, these two types of photosystem have a similarity with the bacterial type photosystem from different organisms. So these two types of photosynthetic bacteria are called as type 2 phototroph, including purple bacteria, and also type 1 phototroph, including green sulfur bacteria and heliobacteria. In type 2 phototroph, reaction center constitutes a cyclic electron transport with cytochrome BC complex, and this reaction center is similar to the photosystem 2. On the other hand, in type 1 phototroph, reaction center constitutes a linear electron flow to reduce the ferredoxin with cytochrome BC complex. And in oxygenic phototroph, like cyanobacteria, these two types of reaction center are combined in one cell. However, this is not enough to oxidize the water and reduce the ferredoxin and LDPH. So this slide shows the energy diagram of the redox cofactor in type 2 reaction center and the type 1 reaction center, and also photosystem 2 and photosystem 1. So P870 and P680 and P700 and P840 is the reaction center chlorophyll that is excited by light and reduce the acceptor side molecule and oxidize the donor side molecule. You can see the similarity in the acceptor side in type 2 reaction center. So in type 2 reaction center, quinone is a mobile electron carrier that transfers the electron to the cytochrome complex. On the other hand, in type 1 reaction center, electrons are transferred to iron sulfur cluster and then ferredoxin and then NADP. So similar electron transfer can be observed between the type 1 among the type 1 reaction center and PS1. A striking difference is here. You can see that large positive shift of peak reaction center chlorophyll in PS2, almost 1.0 millivolt, and this positive midpoint potential is essential to oxidize the water that have a midpoint potential around 0.83. And because of this positive shift, now cyanobacterium can oxidize the water and then reduce the ferredoxin and NADPH to make the oxygenic photosynthesis. Because of this special character of water oxidation mechanism by PS2 is one of the hottest topics in plant science. And this is a crystal structure of photosystem 2 complex from cyanophilic cyanobacteria. Structure is resolved at 1.9 angstrom resolution, and this paper is published in Nature 2011. And you can see that number of membrane spanning subunit and also several membrane extrinsic subunit in photosystem 2. And the catalytic manganese cluster is located on the cycloluminal side of photosystem 2 complex. Here I'd like to introduce a simple guide to the resolution of protein structures. I hope it will help you to evaluate the structural paper. So above angstrom resolution, only secondary structure can be assigned, while protein fold can be determined around 3.0 angstrom resolution. To investigate the chemical reaction at molecular level, the resolution should be around 2 angstrom. Of course, higher resolution enables us to make more accurate discussion about the reaction mechanism. And this slide shows the electron density map around the catalytic manganese cluster in PS2, reported by UK group and Japanese group. You can see that at the 3.4 angstrom resolution, it is difficult to assign the structure of manganese cluster. On the other hand, at the 1.9 resolution, you can see the clear electron density of each manganese atom. In fact, this is the first report showing the structure of the catalytic site of PS2. Besides, number of water molecules shown in red spheres are observed in the structure, and one of then should be the substrate water to be oxidized and evolve oxygen. Okay, so it is known that manganese cluster in pH2 takes 5 redox intermediate state called S-state cycle. The left panel shows the famous experiment by Joliot showing the 4-flash oscillation of oxygen evolution. So this is so-called Koch cycle. Inspired by this data, Koch proposed a 5 redox intermediate of the manganese cluster, and this is a current model of water oxidation and oxygen evolution. Among these S state, S1 state is a stable state of the manganese cluster in the dark. So that means the structure determined by Chen and Kamiya group in Japan is the structure of S1 state. But to elucidate the mechanism of water oxidation, we have to know the other intermediate redox state of the manganese cluster. But it is quite difficult to get such intermediate structure by conventional X-ray analysis, crystal structure analysis. However, recent structural analysis using X-ray free electron lasers make it possible to obtain the snapshot of the reaction. Now there are big competition to determine the water splitting mechanism. Here I'd like to show a video showing how XEL used to get the snapshot of the S-state cycle.
You're probably familiar with photosynthesis, the process by which plants, algae, and other related organisms use light energy to convert carbon dioxide and water into sugars and oxygen. This incredibly important multi-stage process is not only responsible for providing most of the energy necessary for life on Earth, but also for maintaining the oxygen in our atmosphere. Now, using the high-energy X-ray laser pulses produced by SLAC National Accelerator Laboratory's LINAC Coherent Light Source, scientists at Lawrence Berkeley National Laboratory, in collaboration with scientists at SLAC and Stanford University and collaborators in Germany, Sweden, and the UK, have developed a powerful new way to study exactly how photosynthesis produces the oxygen that we breathe. These researchers have taken extremely high-resolution images at different stages of the process at room temperature, allowing them to search for how and where the oxygen is produced under real-life circumstances. The researchers specifically studied the protein complex photosystem II, which produces oxygen from water in a small region of the protein during photosynthesis. First, room temperature crystals of the protein were introduced as either a liquid jet or as tiny drops made using sound waves on a moving tape. As the crystals moved forward, they were hit with pulses of visible light, which triggered different stages of the photosynthetic process depending on the number and timing of the pulses. Finally, the drops were hit by powerful X-ray pulses from the LCLS. By looking at how the X-ray light scattered onto two different kinds of detectors, the researchers were able to obtain extremely high-resolution structures of the oxygen-producing region of the protein, giving them new insights into where the bond between oxygen atoms might form from two water molecules, and where it might not. Specifically, by watching where ammonia molecules, which are analogs of water molecules, bonded to the region, they were able to rule out some possible locations where the oxygen bond might form. If the ammonia bonded to a location and oxygen was still produced, then that site was not a likely location for water or the formation of oxygen. Using this and related data, the researchers were specifically able to raise questions about two of the most popular locations, demonstrating just how powerful this technique can be. While they haven't found the answer yet, the researchers hope that by continuing to improve this process and gathering more and more data at different stages of photosynthesis, they can gradually zero in on the exact way that oxygen is produced from water within photosystem II. Understanding how this system works could lead to a revolution in the development of artificial photosynthesis, solar fuels, and more. But most importantly, it could finally help us understand the atomic origins of the oxygen in the air we breathe every day.
And based on this structure, they propose the possible mechanism of OO bond formation to make the molecular oxygen, and also propose the proton transfer pathway from the OEC to the reaction center. And finally, very recently, several months ago, American group I showed in video, reported the structure of S0 and S1 and S2 and S3 state. Based on this structure, they propose that water 3 bound to the calcium is a substrate water to be oxidized. And they also propose the OO bond formation, but still there are 3 models. But I think this model will be determined in near future. Okay, so let's move to the second part about the acquisition of photosynthesis via symbiosis. So as already mentioned, oxygen in our atmosphere is produced by oxygenic photosynthesis organisms such as cyanobacteria, algae, liverwort, and hive plant. And chloroplasts in eukaryote have been evolved from symbiotic ancient cyanobacteria. This first symbiosis is called as primary symbiosis. But in addition to this primary symbiosis, there are another type of symbiosis that's called secondary symbiosis. In secondary symbiosis, number of different type of eukaryotic protozoa engulfed the eukaryotic algae and made them as a cellular or lamellar. And by this secondary symbiosis, number of different type of algae have been developed during the evolution. And furthermore, there are tertiary or serial secondary endosymbiosis, and this make the evolution of algae very complicated. Panel E shows the cellular structure of cryptophyte type alga, and this plast of this alga contains 4 layers of membrane around the plastid. So that means that one is a phagosomal membrane, and the second one is a plasma membrane from the host cell. And this plastid also have the inner and outer membrane derived from the algal plastid. The nucleus of alga is become to the nucleomorph, and almost all genes are transferred to the host nucleus during the evolution. And surprisingly, this secondary symbiosis process is still ongoing in some algae. This slide shows the image of some alga called hatena. Hatena means question mark in Japanese. So this the flash of this alga have 4 layers of membrane, typical to the the plastic generated by secondary symbiosis. And the interest point is that define the cell division. Plastic derived from algae is transferred to the only one of the daughter cell, and another daughter cell become a predator to obtain the new algae and make them as a plastic. So this suggests that the acquisition of oxygenic photosynthesis is still ongoing at present. And another type of the acquisition of oxygenic photosynthesis is a process called kleptoplasticity. So this sea slug is brown in larvae and juvenile stage, while it eats some specific algae and obtain the plastic and make the plastic into their own cells. This acquisition of plastic is absolutely required for its development. And you can see that complete green color of the adult sea slug. So it is still unknown that how this sea slug maintains the exogenous plastic for a long time, but it is apparently beneficial for this sea slug to obtain the oxygenic photosynthesis. Then let's think about the human photosynthesis in serious way. I think there would be two major problems. First is the solar energy is unlimited, but quite dilute. So solar energy reaching to the surface of the Earth is about 1 kilowatt per square meter. That corresponds to the about 860 kilocalorie per square meter per hour. And body surface area of adult man, like me, is 1.7 square meter, but we can use only half to face the sunlight. And energy conversion efficiency is at most 5%, but usually 1% in C3 plant. So total gain of one should be, this one should be 0.05. Is about 37 kilocalorie per hour. But usually we require the energy per day about 2,000 kilocalorie. So that means that we need a large surface area to get the sufficient energy to survive. However, we cannot move with this large body surface area. And another problem is the oxygenic photosynthesis can be very harmful. So there is a massive electron flow on the cycle membrane of chloroplast, and electron can be easily transferred to the oxygen to generate the reactive oxygen species and ROS, and this can damage the host cell. To cope with this problem, plants have precise control mechanism light harvesting system, and also plants accumulate various antioxidants such as ascorbate, carotenoid, tocophenol, or flavonoid, and so on. So when we think about the two major problems of oxygenic photosynthesis, we have to have large surface area, and we have to accumulate the various antioxidants. So final life form should be like this. So that means that there are benefits as well as drawback to do oxygenic photosynthesis. And that means that to do photosynthesis is almost to be a plant. Okay. So And in the final part, I'd like to talk about the protection mechanism of photosystems against excess light environment. So when we think about the change of the living environment during the evolution, each photosynthetic organism should adapt to those living environment, such as change in the light quality and quality, and also they have to cope with the stresses caused by direct sunlight or dehydration. To cope with these problems, there are several adaptive mechanisms. For example, changes in light harvesting system and the pigment can utilize the different wavelengths of color of light. And in addition, plant developed dynamic adjustment system of the light harvesting capability. In particular, dissipation of excess light energy as heat, so-called non-photochemical quenching, NPQ, is very important to cope with the excess light irradiation. Then, how plants respond to the excess light? As I told you, during the photosynthetic electron transfer, protons are translocated into cyclolumen. And there is additional type of electron transport around the photosystem 1 that also enhances the translocation of the proton into cyclolumen. The key factor induced the photoprotect mechanism against excess light is the accumulated proton, resulting in the delta pH across the membrane. So under the excess light condition, the accumulated proton in cyclolumen downregulates the oxygen evolving reaction and also electron transfer at cytochrome B6F. This prevents the over-reduction of photosystem 1 and also prevents the generation of reactive oxygen species at the terminal side of photosystem 1. However, under this condition, plastoquinone pool is fully reduced and highly oxidized P680 plus remains as in the oxidated state. This condition is very dangerous for photosystem 2 and it should cause the oxidative damage around photosystem 2. To prevent the overexcitation of PS2, plants have various mechanisms to protect the photosystem 2, such as thermal dissipation of excess light energy, so-called non-photochemical NPQ, and also state transition system is balanced the excited state of the PS1, 2, and PS1. And finally, damaged PS2 is effectively removed by the PS2 repair system. And in next part, I will explain each protect mechanism more in detail. So in thermal dissipation of excess light energy, NPQ, one of the cytochrome membrane protein named PSBS have an important function. As shown here, PSBS, shown in blue bars, modulates the structure and function of PS2 antenna with the light harvesting protein. And protonation PSBS under the high delta pH condition. Conformational change occurs by the protonation of PSBS. And this causes the detachment of LSC2 from the PS2 and also causes the aggregation of LSC2 antenna to induce the non-hot chemical quenching. So PSBS has a central function to regulate the largest light harvesting capability of LSC2. Besides, an NPQ, non-hot chemical quenching, also depends on the xanthophyll cycle pigment bound to the LSC2 antenna protein. As shown here, a large delta pH, high delta pH activates the activity of beta-xanthine epoxidase, VDE, and VDE converts the beta-xanthine to anthraxanthine and finally zeaxanthin. And this zeaxanthin carotenoid have a function to relax the excited chlorophyll to the ground state and dissipate the light energy as a heat. And under the low light condition, low pH condition, zeaxanthin back to the beta-xanthine and chlorophyll, excited chlorophyll transfer the energy to the photosynthesis to generate the sugar. So PSBS together with xanthophyll cycle controls the thermal dissipation of excess light energy in plant. And another mechanism is state transition. So this cartoon shows the PS2 dimer surrounded by the antenna system, and under the overexcitation condition PS2, a protein kinase activated to phosphorate the LSC antenna protein, and phosphorylated LSC protein move to the photosystem 1 and prevent the overexcitation of PS1 and also oxidize the PS1. So this is also important to balance the exciting state between PS2 and PS1 and green algae state transition system preferably. The final protection mechanism is PS2 repair cycle, and when plant exposed to the highlight, PS2 is prone to be damaged, especially the reaction center D1 protein is damaged. And this inhibition is called photoinhibition, and this photo-damaged D1 protein should be removed by the protease, and newly synthesized D1 should be inserted to the reaction center. And the plant have very efficient repair system for PS2, and this repair system is essential for plant to be survived only even under the normal growth condition. So in summary, plant have various systems to support the photosystem function, such as NPQ, non-hot chemical quenching, and in NPQ, PSBS and VDE control the light harvesting capability, and this is determined by delta pH across the cycle membrane. And in state transition, protein kinase called STN7 reduce the LSC2, and phosphorylated LC2 move to the photosystem 1. And under the normal condition, protein phosphatase called TAP38 dephosphorate LSC2 and LSC2 back to the PS2 to activate the photosynthetic water oxidation. And finally, PS2 function is maintained by the PS2 repair cycle. Okay. So, in the rest of time, I'd like to introduce the work of our group done by the Miss Saki Yasuhara, a master's student. And she investigates the role of various photoprotective mechanisms against prolonged PS photo emission. She used Arabidopsis PSB01 mutant as a model system for PS2 photo inhibition and investigated the role of various photoprotective mechanisms in this PSB01 background. And she also analyzed the non-photochemical quenching in PSB01 using the fluorescence lifetime imaging microscope. So this is the Arabidopsis PSB01 mutant she used as a model for the PS2 photo emission. This mutant defective in PSB01 protein that stabilizes the catalytic manganese cluster in PS2. As shown here, because of the PS2 photo emission, PSB1 shows the pale color leaf and retarded growth even under the normal growth condition. And what she did is she made a double mutant of PSB1 with other photoprotective mechanism such as PSBS or cyclic electron transport that produce the delta pH to induce the non-photochemical quenching. And this is the visible phenotype of PSB01 and as a double mutant. And you can see that the growth of PSB01 is severely reduced because of the PS on photo emission. And when we compare the growth between the PSB01 and the double mutant, you can see that double mutant of PSB1 and PQ4 that lacks the PSBs shows a severe growth retardation when compared with the PSB01. This suggests that PSBS is essential for the protection mechanism against PS2 photo emission. Then we investigate the allocation of absorbed light energy in PS2 by using chlorophyll fluorescence analysis. So we can estimate the allocation of absorbed light energy by measuring the chlorophyll fluorescence. And energy can be divided into 5 MPQ with a fraction for the non-photochemical quenching and phi2 photochemical quenching that is used for the photosynthesis, and also basal thermal dissipation as a fluorescence. This model is based on the Hendrickson reported in 2004. And this slide shows the changes of allocation of absorbed light energy in PS2 under the light condition. You can see that in wild type, pi FD basal thermal dissipation is almost stable. On the other hand, the fraction in NPQ is a little bit high at the starting point of the light emission, but it becomes smaller. In PQ4 lacking PSBS, you can see that increase of phi FD, but still phi2 is maintained. But on the other hand, in PSB01 mutant, you can see that large increase of non-photochemical quenching that will be important to protect the photo-damaged PS2. And when we made the double mutant of PSB1 with PSBS, you can see that very much increase of some basal thermal dissipation, and this basal thermal dissipation would be dangerous for plant and retards the growth of PSB1 mutant. On the other hand, the contribution of other cyclic electron transport pathway is not so important for the growth of PSB1 mutant. Then Yasara-san also investigates non-photochemical quenching in PSB01 using chlorophyll fluorescence lifetime analysis. In this analysis, he measured the fluorescence lifetime with the film with a laser power of different with a sequential different laser power, and then plants are treated with highlight and relax the NPQ profile, and again we measure the chlorophyll fluorescence lifetime with a single photon counting. And this slide shows the typical image of wild-type leaf using fluorescence lifetime imaging microscope. You can see that chloroplast, the fluorescence of chloroplast is a mesophyll cell, and location of chloroplast is almost similar between before and after the highlight treatment. And this is the image of the leaf of PSP01. You can see, again, you can see no difference between the before and after the highlight treatment. But when we measure the chlorophyll fluorescence lifetime, there is a difference. So in the wild type, when we include the laser power, we can see that increase of chlorophyll fluorescence lifetime. That means a reduction of electron transport system under the high laser intensity, in the higher laser intensity. On the other hand, when we treat the plant with highlight, this increase is suppressed, indicating that induction of non-photochemical quenching in the wild type. On the other hand, in PSP1 that have damaged PS2, fluorescence lifetime is much longer than wild type. This indicates that energy transfer from light harvesting complex to PS2 is inefficient in PSP01. But interestingly, when we increase the intensity of laser power pulse, you can see the decrease of the fluorescence lifetime under the stronger laser pulse. This indicates a strong induction of non-photochemical quenching, and also after the highlight, this induction of NPQ is enhanced in PSB1 mutant. So this CH1 mutant is a mutant lacking chlorophyll B and lacking the LSC2 antenna system. But so in this CH1 mutant, we don't see any difference between the before and after the highlight treatment. So that means that this chemical quenching observed, especially observed in PSB1, depends on the light harvesting system that have a chlorophyll B. Then we measure the chlorophyll fluorescence lifetime in PSB1 and MEQ4, and you can see that no induction of the shortening of the chlorophyll fluorescence lifetime. So that indicates that strong NPQ in PSB1 mutant is totally dependent on the NPQ for PSBS protein in PSB1. So okay, so this is a summary of Yasahara-san's work. So first, NPQ in PSB1 requires PSBS, and this would be important for the sustaining its growth. And the longer chlorophyll fluorescence lifetime in PSB1 would be due to the detached LSC2, which cannot transfer light energy effectively. And PSB1 showed the chlorophyll fluorescence lifetime and the high exciting laser pulse, and this phenomenon was enhanced and long-standing after the highlight treatment and also requires PSBS. And these responses involving PSBS would be beneficial to dissipate excess light energy when the linear electron from PS2 is severely suppressed under the prolonged photo inhibition. Okay, so today I talked about the protection mechanism of against the excess light energy, but there is a emerging attractive vision to engineer the crop with improved photosynthesis efficiency. It is estimated that by doing the optimization of light harvest system, plant could grow with 15% increase of photosynthetic activity. That may result in the increase of crop yield. And this table shows the potential target for the enhancing of photosynthesis, including light harvesting system or photochemistry itself, or production of ATP, or alternative electron flow to remove the excess electron in safe way. And furthermore, as I told you, photoprotection mechanism, including non-photochemical quenching and its repair, is also the target of genetic manipulation. So there is an exciting possibility to manipulate the photoprotection mechanism and to enhance both stress resistance and photosynthetic productivity of crop plant. And today, Ms. Kristen Cho introduced a paper related to this topic published in Science in 2016. I'm not going to explain it in detail, but because she will explain the detail later, but the key concept is that keeping track of sunlight by controlling the NTPQ can enhance the plant productivity. So this is today's take-home message from my lecture. First is I talked about the basics and the evolution of photosystems, and I showed that mechanism water splitting reaction is almost elucidated in very near future. And I also talk about the acquisition of photosynthesis via symbiosis. And I also talk about the human photosynthesis. And there is a benefit and risk in the oxygenic photosynthesis. And finally, I talked about the protection mechanism of photosystems against excess light environment. And suggests the possibility of photosynthesis that photosynthesis can be improved in future environment. So that is my lecture. Thank you.
Okay, let's take a 5 minutes break before the student presentation. And does anybody have any questions?
One question, I know this is outside your field, but I was intrigued by the symbiosis, especially the clastoplastidity of the sea slug. And most organisms recognize foreign versus self. Has anyone looked at cell surface receptors to see how this kind of symbiosis works? So the sea slug engulfs the algae and then discards this algae nucleus and maintains only the plastid.
Yeah, so in case of this sea slug, they take only the plastid, not maintain the whole cell. Yeah, and there is a report indicating that this sea slug have some algal gene to maintain the chloroplast. Yeah, but there is still debate about the process to maintain the chloroplast.
And as we know, in plant evolution, so many genes have been moved from the plastid genome into the nucleus. How does the plastid maintain the ability to photosynthesize?
Very important question. So the researcher invests the genome of this sea slug, and they suggest that some chloroplast gene is exist in the nucleus of this C slug, but not everything. Yeah.
One question from Kyoto student.
Thank you for your lecture. So I just have a small question. So at the last of your talk, you talk about by modifying, you can increase the efficiency of photosynthesis. So is there any plant already existing naturally, so that it may have much higher efficiency than the other plants?
By genetical modification or
So naturally
natural
existing plants. Did you find any?
Yes. So of course, the productivity of plant is different between the species. For example, you may know the C4 plant.
Oh yeah.
Yeah, that have large productivity when compared to the C3 plant, but under some special condition. So the plant productivity is totally depend on the growth condition. So some plant is very good at the higher light intensity, but some plant is grow well under the cold condition. So there is some spectrum the plant prefer.
Oh, I just guess maybe if you could find any fascinating plants existing in the nature, so you found, oh, this plant have so high efficient in photosynthesis, and you can use the gene from this plant.
Yeah.
I just thought.
Yeah, actually, there is a research project to introduce C4 genes into the C3 rice, but it's quite difficult to obtain such a complicated mechanism.
Okay, thank you.
Okay, let's take a 5 minutes break.
Yeah, I think we can start.
Hello, everybody. My name is Crystal Chow, and I'm from Kyoto University. And today I'd like to introduce this paper that was published in Science titled Improving Photosynthesis and Crop Productivity by Accelerating Recovery from Photoprotection. So as you may know, the world population has been increasing, and along with it, the demand for food has been projected to increase as well. This graph here shows the projected yield, which is shown in the solid line, and the projected demand for some key crops, including wheat and rice. And unfortunately, at the rate we're going right now, the demand is going to be greater than the amount we can produce. So which is why it is important to start thinking of ways on how we can improve crop yields. So one method to do that would be to increase the efficiency of photosynthesis. And I'm just going to go over this really quickly because Dr. Ifuku went into it in a lot of detail already. But basically, photosynthesis is the process of converting CO2 and water, along with light, into sugar and oxygen. And this process occurs in the chloroplasts of plants and can be divided up into light-dependent reactions, which occur in the thylakoid, and light-independent reactions. So light-dependent reactions produce NADPH and ATP, which are used in the further reactions to eventually produce the sugars. The membrane of the thylakoid is where the light-dependent reactions occur. This is an electron transport chain with two key photosystems, photosystems 2 and photosystems 1. And photosystem 2 is the protein complex responsible for splitting water. And this process generates protons into the thylakoid interior, which then drive ATP synthase to produce ATP. So these are very important components for the cell and are damaged in high light conditions, which is why plants have developed ways to protect these systems during times of high light. So in response to high light conditions, the plant undergoes, so when a molecule of chlorophyll is excited, it can undergo either one of three possible fates, which are in competition. Either it can emit fluorescence, it can undergo photochemistry, or if there's any excess energy, it can dissipate it through heat by non-photochemical quenching. And non-photochemical quenching itself can be divided up into several components. So for the purpose of this presentation, I'll be focusing on QE, which is the energy-dependent quenching that involves the acidification of the thylakoid membrane. And as we heard earlier, this causes protonation of the subunit of photosystem 2, PSBS. The QZ component is the zeaxanthin-dependent quenching component, and this involves the pigments that the pigments of violozanthin and zeaxanthin. So violozanthin and zeaxanthin are pigment molecules known as xanthophylls, and these are part of what is known as the xanthophylls cycle. So the difference between them is the presence of this epoxy group, in violozanthin and these double bonds in zeaxanthin. And the conversion between them is mediated by the enzymes VDE, violozanthin deepoxidase, and ZEP, zeaxanthin epoxidase. So how do PSBS protonation and zeaxanthin promote quenching? So I think the exact mechanisms aren't completely characterized yet, but as we heard earlier, PSBS causes a conformational change to around photosystem 2, and along with zeaxanthin, this is thought to form what is known as a quenching complex that facilitates efficient NPQ. So to summarize, in high light conditions, you get efficient CO2 fixation, and any excess energy is dissipated by NPQ because PSBS is protonated, and the predominant form of the xanthophylls are zeaxanthin. However, once the light conditions switch over to low light, so for example, if a cloud passes over the sun, then NPQ is still active and it is inhibiting photosynthesis, so you get inhibition of CO2 fixation. In order for efficient CO2 fixation under lower light conditions to return, NPQ must relax, so PSBS must be deprotonated, and zeaxanthin must be epoxidated into violazanthin. And unfortunately, this process is relatively slow, so there's a lot of loss in potential photosynthetic efficiency due to having to wait for NPQ to relax. So the authors of this paper wanted to think of ways on how they can increase the rate of NPQ relaxation. And they did this using two strategies. So previously published research has shown that overexpression of the S subunit of photosystem 2, PSBS, increased the rate of induction and relaxation of QE of NPQ. Another approach would be to alter the xanthophyll kinetics, and it was previously found that in Arabidopsis mutants that higher xanthophylл pigment pool had slower NPQ relaxation and induction. So therefore the authors thought that by maybe adjusting the turnover and distribution of these pigments, it might also adjust the rate of NPQ. So in order to test this hypothesis, the authors decided to overexpress the enzymes that convert between the xanthophylls, as well as PSBS in plants, and see what changes that would cause. So here they transform Nicotiana tabacum plants, which are basically the tobacco plants, with Arabidopsis, VDE, ZEP, and PSBS. And they selected two progeny with single tDNA integration, and one with double tDNA integration, and then allowed them to self-pollinate to obtain homozygous T2 progeny, which they used for their further experiments. First, the authors decided to check on to see the expression levels of their transformed genes. So figures 2A, C, and E show the mRNA levels in the VDE, PSBS, and ZEP in the wild type and VPZ plants, respectively. And the gray bars here indicate the endogenous native levels of the genes, and the red indicates the Arabidopsis transgene. So here you can see that in all of the VPZ plants, they have higher mRNA levels for each of the three genes. And in figures B, D, and E, we can see that this translates into increase in protein levels as well. So here they show that the VPZ plants have higher VDE, ZEP, and PSBS mRNA and proteins. So next, they wanted to know whether what effects these changes have on the rates of NPQ relaxation as well as photochemistry. So to do so, they used a technique known as chlorophyll fluorescence imaging, which was also briefly introduced in the lecture. So as mentioned earlier, light-excited chlorophyll can undergo one of three fates, which are in competition. Therefore, information on NPQ and photochemistry can be obtained by measuring the fluorescence from chlorophyll under certain conditions. So this is just a schematic diagram of a typical fluorescence trace that is done to figure out some of these parameters. So first, plants are left in light-limiting conditions, and under these conditions, there is neither quenching occurring nor photochemistry. And after first a low intensity of light is administered, which is called the measuring beam. So this amount of light is enough to induce fluorescence, but not quenching or photochemistry. So this gives a minimal amount of fluorescence. Next, a saturating pulse of light is administered. And what this does is that it transiently closes all of the PS2 photocenters that prevents photochemistry from happening. And since the plants are adapted to dark conditions, there's also no quenching happening. So this FM value here would be the maximal amount of fluorescence that can, so the maximum possible fluorescence. The plant then can be moved under actinic light, which is just a term that means light conditions where photochemistry can occur. And then you can see an initial sort of increase in fluorescence, which then decreases due to photochemistry and quenching. And eventually it will reach a state where all of those three are balanced, called here denoted by F prime, known as steady state fluorescence. If you administer another pulse of saturating light under these conditions, you can get another peak here, which you can see is quite a bit lower than the one that was measured in the dark. So once again, the saturating pulse of light would prevent the contribution from photochemistry. However, so this decrease would be due to non-photochemical quenching. So to calculate how much NPQ is occurring, we can use these two values and subtract them and then normalize them to obtain NPQ. Similarly, the relaxation can be measured by returning the plant into dark conditions and then periodically administering the pulses to see how long it takes before it reaches the maximal fluorescence. In order to calculate the operating efficiency of photosystem to quantify the amount of efficiency of photochemistry, we can use the FM prime, which is the fluorescence due to fluorescence in the presence of NPQ, but not photochemistry, and then take the steady state fluorescence, which is fluorescence when there is both photochemistry and quenching. And by subtracting these, you can get only the contribution of photochemistry. So using these sorts of measurements, the authors first decide to investigate the rate of NPQ relaxation between the wild-type plants and the VPZ plants. So the authors exposed the plants to fluctuating light and then allowed them to relax in the dark. The graph on the graph over here shows the VPZ plants in red and the wild-type plants in gray. And you can see that here, in the VPZ plants, you get faster decrease or relaxation of NPQ when compared to the wild-type plants. Here in figure 3b, the authors investigated the net CO2 fixation rate. And they first administered a high intensity beam of light and then tracked the CO2 fixation over a course of about 150 seconds. So in both the wild type and the VPZ plants, you get a sharp decrease in net CO2 fixation after the administration of the light. However, over the next 150 seconds, you can see that the VPZ plants reach higher levels when compared to the wild type plants. And this averages out to about a 9% increase over the time frame. Next, the authors compared the performance of the plants under either steady state conditions or fluctuating light conditions. And here you can see on the, in figures A, C, and E that under steady state conditions, the CO2 fixation rate, PS2 operating efficiency, as well as NPQ aren't actually different between wild type and VPZ plants. However, once these plants are put under fluctuating conditions, there is increased fixation, CO2 fixation, as well as PS2 operating efficiency in the VPZ plants, and you can also see there is lower NPQ. Next, the authors took a look at the composition of the xanthophyll pool under various light conditions. So in the dark conditions where there's no non-photochemical quenching occurring, in both the wild type and the VPZ plants, the xanthophylls are in the deepoxidated form. They're on the epoxidated form, I think, violazanthin. However, quite interestingly, once the plants are exposed to moderately high amounts of light, you can see that the VPZ plants retained more violazanthin in comparison to the wild-type plants, where the pool has shifted towards to have about equal amounts of both violazanthin and zeaxanthin. A similar trend is also seen when the plants are put under fluctuating light. So here, once again, the VPZ plants retain a higher percentage of violazanthin compared to zeaxanthin, while in the wild-type plants, it's shifted more towards zeaxanthin. So how do all of these changes actually affect crop yields is the big question. So here in the next experiment, the authors decided to grow plants under field conditions. And then at the end of about 22 days, I believe, the authors investigated how big the plants grew. So here they looked at the dry weight of the plant, the leaf area, and the plant height. And all of this data here is shown relative to the wild-type plants. And so here you can see for all of the metrics that they looked at, all of the VPZ plants showed increased size when compared to the wild-type plants. So to summarize, the authors sought to reduce the time needed for NPQ relaxation in a couple of ways. So first, they overexpressed PSBS, which is the subunit that is protonated during non-photochemical quenching. And they also tried to alter the kinetics of the xanthophyll cycle by increasing the amounts of VDE and ZEP. And together, this may facilitate a faster reversion from the complex that causes quenching to one which can efficiently perform photosynthesis. So in doing so, they found that their VPZ plants do indeed have increased NPQ relaxation. And as a consequence, this also leads to increased photosynthetic efficiency under fluctuating light conditions, and eventually also leads to larger plants. So although this research was performed in tobacco plants, the authors think that maybe this strategy could be applied to other crop plants, and that may eventually help increase yields as well. So that's the end of my presentation. Thank you for listening.
Thank you. Does anyone have any questions? No.
Yeah, thank you very much for a good presentation. So it looks very, very promising, but are there any drawbacks? Or...
Yeah, so I don't think the authors really mentioned any drawbacks, but there was also something that I was wondering about, as well as with a question earlier on if this sort of modification leads to larger plants. I was thinking that it would lead to some evolutionary advantage, but would there be a reason as to why we don't see plants with elevated levels in these proteins already? Like, would there be some sort of downside? It's something that I was wondering about as well, but it's not really touched on in this paper, I think.
Hello. I'm the student from NTU, and I've got a little question. Is that in the introduction of the non-photochemical quenching, the MPQ, and I'm quite curious about what is the QZ, which is the opposition of violatization to zethane. So that part I'm not get the picture. So can you explain more about these parts?
So you were wondering about this
The QZ part
over here?
Yeah.
Yeah. Sorry, I guess I might have not explained it well, but the QZ part is pretty much is about the xanthophyll, the cycle, the shift from viral towards zeaxanthin, which helps in the process of quenching the excess energy.
Okay, so thanks to your explanation.
Thanks for your presentation. It's clear, but still have a question. Why did they do the fluorescence test? Is there any association in the MPQ and the CO2 fixation?
So CO2 fixation would be a way to see how much photosynthesis is occurring, because the process involves taking CO2 to produce oxygen. Sorry, was that what you were asking about?
I'm just curious about why did they do the fluorescence test?
So fluorescence imaging allows them to, because the fluorescence signal would be reduced when either NPQ or photosynthesis is occurring. So by measuring changes in the fluorescence, you can calculate the contribution of NPQ or photochemistry.
Thank you.
Hi, this is from NTU side. So if the fluorescence and the MPQ, you mean the decrease of the fluorescence indicate the MPQ are increasing. That means the sum are the same. If the sum are the same, why is there increased MPQ can improve the photo damage? I mean, if you increase MPQ, then I guess the fluorescence will decrease. Then the total beneficial should be equal. Or am I making a wrong assumption?
So what NPQ does is that it causes the excess energy to be dissipated as molecular vibrations. So yeah, when you have increased NPQ, you would, I guess in some cases, you'd have decreased photochemistry as well. So usually you only want it to be active when there is excess light energy. However, as shown earlier, it takes a while to relax. So sometimes even in lower light conditions, it's still active, and this is not beneficial to the plant because it impedes photosynthesis.
I think one earlier question addressed CO2 fixation, and that's the dark reaction, so unrelated to the light reaction. So I'm just wondering if they're exploring kind of feedback mechanisms between NPQ and how that might affect Rubisco or the dark reaction. I'm just suspecting it might be a little more complicated than this.
Yeah. So I don't think they explain too much about the dark reactions in this paper, but I think they're mainly looking at it as just as more evidence that photosynthesis is occurring in these plants, and that if you get increased CO2 fixation, then these plants might be more efficient in doing more photochemistry. I think that's the main reason why they're looking at.
So I think you used the tobacco plant, but I think it's already domesticated. So are there any differences between wild species and those species used in agriculture or something?
I'm not too sure about that, but I suppose this would be something that the authors are keen on exploring further. So this is just an initial experiment, but it has promising results which might be able to be used in more real-life conditions.
I have an answer for your question. So actually, the expression level of PSBS is different among the plant species. And the level of PSBS is directly correlated with the ability to induce NPQ.
Are there any more questions? If not, so I think it's the time for closing. Thank you very much.
Thank you. See you next week.